So, hello everybody, my name is Giuliano Scarcelli. This is a little presentation of what we do in my lab. I am a physicist, a quantum optics experimentalist as a training, but over the past 10-15 years I've mostly been working in biomedical optics. So the focus of our lab is mostly to use the bio universe and as an excuse to do interesting optical and light matter interaction experiments. So this first slide that I have is the general themes that are touched in the lab, and then I will have a more detailed description of the majority of the efforts that we do. So one of the things that we are working on is the ability to imaging through turbine media. This is very interesting problem because, as you may know, light from a practical standpoint, in the biomedical world, light is not very useful unless you use endoscopes, because light does not penetrate into tissue. On the other hand, light is not lost. Most of the light is not absorbed, but it's just scrambled by diffusing. So by studying this diffusion of light, we are studying and we are starting to develop ways to basically descramble the light and get information of objects behind visually opaque samples. So this is one example in which we had I think it was a 3 millimeter chicken tissue breast, and this was the object, goat herpes. And here you see what your camera would just get out of that, and this is the reconstruction we were able to do. Another interesting topic that we are working on is bio-optics in several different forms. The basic idea is that we want to make optical components that are biocompatible or made of biological tissue, biomaterials and stuff. So in this particular experiment, we basically made polymers that are biodegradable, and you can guide light through them, and the demonstration was that you could now send light deeper into tissue through this waveguide. Recently we have done another cute experiment that we are now working on more and more, in which we are basically changing the temperature of tissue transiently, and that changes the optical properties of the tissue, and so we can do that to our advantage in order to make the tissue itself behave like an optical element. Another topic that we are very interested in is using resonators. Resonators are a very familiar tool in non-biomedical optics. And the reason why they are interesting, right, resonators are like lasers, right? So the reason why they are interesting because by going on resonance, you become very, very sensitive to any changes of the surroundings. So we're using the property of the resonator to create better sensors for forces that cells might apply. This is a particular demonstration in which we put a resonator, and then on top we culture cells, and so when cells move the substrate, we have a very, very precise way to characterize the force that the cell applies. The vast majority of our effort is about photon-phonon interactions. This is a phenomenon that was first described by Brillouin in the 1920s, has been used in material science for a long time, but Brillouin spectrometers are mostly slow, and so they cannot be used in biology. We have done a lot of improvements on the spectrometer, and one of the biggest directions that we are pursuing in our lab is to make them better and better and better, faster and faster and faster. The payoff from the point of view of application is dramatic, because you can see that these Brillouin maps here encode with color something that is proportional to the stiffness of material. So you basically get the ability of mapping the stiffness, the modulus of material, with high resolution and without contact, which is something that you cannot do. Now, I don't know if this audience is interested in this, so there is a lot of biochemical application of this process, because the property of tissue from a biomechanical standpoint and the property of cells from a biomechanical standpoint have been known to be very important. So in this respect, when we develop our instrument, we sort of know already that this instrument is going to be fairly successful from an application standpoint. So let me guide you a little bit through this a little bit more. So what we basically do is that we shine light into a material, and light interacts with an acoustic phonon inside the material and comes back scattered at a slightly different frequency. So now, if you are familiar with Raman, this is similar phenomenon, but just much smaller shift because the interaction is spontaneous acoustic phonons. From a physics standpoint, you can basically understand this as if you're creating a Bragg diffraction, because with the acoustic waves you're creating a modulation of the refractive index inside the material. So that's what a Bragg diffraction is. And then there is going to be a Doppler shift, because this is a traveling acoustic wave. So that's why you get a shift at a given angle and of a shift of a given frequency. Now the beauty of this is that if you can measure the shift, then that shift is proportional to the longitudinal modulus of material, and this is a validation that indeed what we measured is correlated to the modulus of material. If you can measure the line width of this resonance, it's basically the viscosity of the material. This is another one of these calibration. So you see, basically from a practical standpoint, you basically are doing high resolution spectroscopy, and you get, based on that, intrinsic contrast mechanisms, so label-free contrast mechanisms that we use as a base for microscopy, that gives you information about the biophysical properties of materials. So we have gotten to the point of doing it so well that we have done by now, I think, 100 patients. This is me being measured by my postdoc, and this is my eye, which is normal and stiff, while in certain patients you basically get this localized softening, which is an indication of disease. We have also created a flow cytometer based on that, in which we can basically measure and a confocal microscope, in which we will basically map the stiffness of the cell or measure a bunch of cells quickly. And this is fairly useful because metastatic cells happen to have very different biophysical property from normal cells. So this could be some way of refining your ability to study and detect metastasis. We are studying various biological processes. For example, tumor cell extravasation is one that we are spending a lot of effort. And we are finding this wonderful, actually terrible characteristic of metastatic cells, which is the ability to change their stiffness during the process of cell extravasation, which basically means that cells are able to adapt the mechanical properties depending on what's needed in the particular process. One other application that we are doing is looking at the back of the eye. You can see we can basically get a nice map of the soft part in the nerve canal, which is next to the very stiff part in the sclera, which is thought to be the reason why glaucoma progresses in the beginning. And we recently got a grant funded to study neural tube closure, this process in development where you have basically the neural folds that close in order to create the beginning of the spine, if you want. And these are Brillouin maps next to OCT maps, and you see that OCT just gives you the structural contrast, so not really information. While with Brillouin, we can basically get wherever the bone is going to be formed, it's much stiffer than wherever the brain is going to be formed. So we're doing a lot of images in this space. So this is a quick outlook of the technology where we are right now. We can do mechanical mapping at high resolution with 3D sectional probability, and I hope you've gotten a sense that we are sensitive to cell and tissue property, mechanical properties that are relevant from a biological standpoint. These in yellow are the things that we are working on, right? The instrument is still slow. It's 2 to 3 minutes per frame. Even though we have improved it so much with respect to the past, still we can only measure mostly transparent material. And I mean, this, in order to truly translate it into a quantitative measurement of mechanics, so modulus, we still need a better understanding. Right now, we mostly rely on empirical calibration, sample by sample. And that transition is one of the most efforts that we do. This is the simplest way to understand how we have gone about this instrument in the past. The x-axis is how fast we can go, and the y-axis is how non-transparent sample can be. So before we started, this was the instrument that everybody used, scanning Fabry-Perot, which could deal with a lot of non-transparent sample, but mostly was going super fast. So we invented a new type of spectrometer that was 100 times faster, and over the course of the years we have improved that to the point that now we are at a point, as I said, that we can do 2-3 minutes per image with the performances that rival the previous instruments. This is the direction where we want to go, right? So we want to have wonderful contrast and super high speed. We are working on that. A graduate student of mine has recently demonstrated we can measure 150 micron chicken breast. So we've gotten to the point of being shot noise limited, in opaque material. So we've gotten to the point that we behave like other microscopes. We are developing now a light sheet version in which we basically measure 300 points at the same time. So it's much faster. And so we have sort of ideas of how to go about a couple of orders of magnitude about this. And so we have a few projects in the lab that are aimed at reaching this spectroscopy, high resolution spectroscopy performance. So this is a picture of the lab and the various collaborators that we have. These are the funding sources. As you can see, they're mostly NIH and NSF. So our grants are generally linked to the application that we are applying our instrument to, but they always contain a technical aim in which we try to do the instrument 10 times faster, 100 times faster. Thank you very much.